Okay, so if you are just joining us on video, the first thing you should do is click on the in-person agenda and watch that 8-minute Anita Sister video. That's what we just finished. So, from your guided notes, we want to talk about a few things. We want to reinforce a few things. And there's like 4 slides that I really want to spend a little bit of time talking about today. And the 4 slides are dealing with like the scientists that kind of got us to where we are, somewhat, I should say, kind of led us to where we are today. So DNA itself isn't a new thing. In the 1950s, it wasn't like we had never heard of this thing called DNA. It was the 1800s, I believe, that is when they kind of discovered DNA. But they weren't sure what it did. They knew things got passed from one generation to the next. They just weren't sure what was the thing that caused those traits to get passed on. So there was a couple different like scientists and experiments that did this. The Hershey Chase experiment in 1952 was one of the ones that kind of got us there. So they knew there was a debate in the scientific community, was it the virus, is it a protein that passes traits on, or is it the DNA that passes traits on? And they didn't really know. So that's what Hershey Chase experiment did. They took a virus that is surrounded by a protein, the genetic information, DNA, is surrounded by a protein. Okay, so they have a virus that has both proteins and DNA. And what they did is they made it, they could make it radioactive, and then they could go through and they could trace it. And that's the idea of doing something that's radioactive, is if I were to inject, they do it with medical like tests. If they want to see if your, gallbladder, right? I think is functioning properly, they'll inject, that might be a dye, that might not be radioactive. But they do sometimes do a radioactive thing. And then they could see how it goes throughout your body, and if it's moving properly, and they can kind of trace that then. So it's something they still use today in terms of radioactive stuff. So they made the protein of one virus radioactive, and in the other virus they took and they made the DNA radioactive. Okay, same viruses, they just changed what piece was radioactive. And then they allowed the viruses to replicate. So this virus had the protein that replicated. Then the future generations, after they let it reproduce, was not radioactive. Okay, it had the same genetic makeup. However, it wasn't radioactive. So therefore, they concluded that protein was not the mode of inheritance, because when they inserted the radioactive DNA, after they let it replicate, the new viruses had the radioactive DNA. So based upon that, they concluded that DNA was the thing that kind of passed traits on in terms of genetics. So that's Hershey Chase. We'll talk more about structural stuff in a little bit. But there's a guy by the name of Chargaff. I always put an extra R in it. Chargas Rule. And the idea for Chargas Rule is that he discovered that in all organisms, no matter what, when we looked at the nitrogenous base, or for just out of laziness, we just say the base pairs. So the A's, the T's, the C's, and the G's. Okay, they're technically called nitrogen-containing or nitrogenous or nitrogenous bases, but we just get lazy and say it's the bases. Okay, so there's 4 of them: adenine, guanine, cytosine, thymine. I don't really care if you know them by name. You just have to recognize that there's letters that go along with them. And Chargaff came up with this proportion. He looked at lots and lots and lots of them, and he noticed that proportionally the A's and T's were the same, and the C's and the G's were the same. So Chargaff's rule, when we talk about that, is for every A there's a T, and for every C there's a G. So memory devices, different ways to do it. In the video they just talked about apples in a tree, right? Apples in a tree, or cars in the garage. Okay. That's how they suggest it. Okay? When I was learning this, it's now become second nature to me. But when I was first learning this information, the way I remembered it is when we talk about how our letters are physically formed, the A's are only straight lines and the T's are only straight lines. The C is curved and the G is curved. So the curved letters go together and the straight line letters go together. That's how I used to remember it. Okay, Mr.
C and G sound the same or very similar, so I always A lot of times I use process of elimination. I know like if there's something, if I remember one, I'll by default know the other. So for me it was, oh, C and G. I like yours, the lines though, better, but, and then by default I know, oh, the A goes with T. CG, that means A goes with T. However you choose to do it.
You just got to remember it, okay? It doesn't, again, lots of different options. You just got to pick one that works for you, but that's what Chargas Rule is. And what we do is we talk about the complement of each other. So the complement of the base A is T. The complement of T is A. The complement of C is G, and G for C. But a lot of times this is what we'll say over and over. For every C there's a G, and for every A there's a T. Or, you know, for every G there's a C, and every T there's an A. But you'll hear us say that a lot, because that's when we talk about complementary base pairing, which we will do a fair amount of. When we talk about DNA replication, when we talk about mRNA making proteins, we do a lot of complementary base pairing. So that Chargas Rule is going to be a crucial thing. In the 1950s, again, we didn't really know structurally what this was. A scientist by the name of Rosalind Franklin did some work with X-rays. And in the 1950s, they didn't know a whole lot about X-rays. It was kind of a newer-ish thing. I don't know when X-rays were actually discovered and done, but they didn't know some of the side effects of X-rays. So when you go and get your, like, go to the dentist, you might get an X-ray. If you think you broke a bone, they'll take an X-ray. And the idea is they are going to take an X-ray because it will help with treatment or preventative or diagnostic purposes. However, there is a risk associated with X-rays because radiation is produced by X-rays, and radiation can lead to cell mutations, cancer. So why do we do that? Because for that one quick second of, hey, we took an X-ray, we know whether your bone is broken, and then we can make some informed decisions based upon that. In the 1950s, they didn't know some of those concerns and those issues. It wasn't uncommon. If you ask your grandparents, maybe, if you have great-grandparents that are still alive, you could ask them. But they used to be, you'd walk into a shoe store. You know how your parents would always like, wiggle your toe, do these shoes fit? I can't tell, wiggle, are you moving your toe? I'm wiggling my toe. You're right, I've had that conversation with my children already many times. So what they had is they had these like X-ray machines that was a live like screen, and people could put their foot under the X-ray and they could look down in the screen and they could wiggle their toes and they could see where their toes are in relation to the end of the shoes. Again, we didn't really know some of the potential health risks associated with that. And it's not just like a X-ray. It was like X-ray while your foot was under there, right? So we don't do that anymore. Rosalind Franklin, why am I saying this? Because she did a lot of work with X-ray diffraction. She took this famous picture of a DNA molecule that has been, this is like the classic Rosalind Franklin picture that you always see associated with her. She did a lot of work with X-rays, and I think it was in didn't mean the year. I believe she was 37 years old when she died, and she died of ovarian cancer, which they believe is linked to the amount of work that she did with X-rays and radiation. So they think actually her work with that led to the cancer that killed her, yeah. So she was a female scientist in the 1950s, and it's not, they weren't considered probably equal. Let me rephrase that. Women in science were not considered equals, and they were kind of looked down upon and not given the credit when credit was due. And that's exactly what happened with Rosalind Franklin, is her work was used but never really credited. And the two people that get a lot of credit for the structure of DNA are the guys by the name of Watson and Crick. And they used Rosalind Franklin's picture to say, hey, it's got the DNA molecule is twisted somehow. How is it twisted? But they never really gave her credit for it. They never said, hey, because of the work of Rosalind Franklin, we kind of got to where we were. At least they didn't initially. And did they later on? I guess I don't know that. In 1962, they won a Nobel Prize for their idea, and they came up with the DNA is referred to as a double helix. So this double helix model, if you can kind of see it, it's not the easiest picture to see, but these are Watson and Crick in like the 50s. There's a metal support rod right here, and the idea is think of it as a spiral staircase, and each like one of these would be a step. So if this is the metal rod, like my iPad stand is that metal rod, there's like a step and then a step, and then a step, and then a step, and then a step, and a step, and a step, and a step, right? They climb upwards. It's a double helix that spirals around it. And you say, if you think about where like my iPad stand was this metal rod, in Rosalind Franklin's picture, that's the metal rod, that's my iPad stand that I rotated around, and that's a stair, that's a stair, that's a stair, that's a stair, that was spiraling up. Okay. So we often hear about Watson and Crick because they were the one that proposed the double helix model of DNA, that is twisted and then is twisted again. Okay, so when we talk about double helixes, that's kind of where we're talking. So in terms of the guided notes, that was some of the big ideas. Some of the other big ideas we will discuss here momentarily when we talk about structural stuff, but that was kind of some of the other pieces of it. Any questions about the guided note piece? There were many people who went through and turned in their guided notes already. However, there are a handful of people who have not yet submitted their guided notes. So, there's 2 sheets that we're going to ask you to grab.
Stack by Morgan and stack up here on the cart. So you have section 2 guided notes, you're gonna need 1 of each. And then there is a packet that looks, the front page, very familiar.
There are 4 of you that did not submit your section 1 guided note. And I could call you by name if you'd like, or do you 4 know who you are? Okay.
Well, I turned mine in.
Jerry?
Yeah.
Okay. Josiah? Yes. Didn't turn in section 1 note. All right, so when we talk about,
Oh, it's not on, that's why.
oh.
Probably did, but.
All right, so when we talk about structural stuff, we are going to look at the thing called DNA structure activity. It's a stable packet, and you might say some of these pictures look very familiar, and that's because they are copied directly off of the Fogel activity. That was one of the other assignments that most of you turned in, with the exception, I believe, of 4. Also, for the same 4 people didn't turn that in either. So we're not going to go through all of this. We really just want to use this kind of as a reinforcement of, hey, we need to make sure we're clear on structural stuff of DNA. So Mr. DeVore circled and highlighted in the upper left-hand corner, that is a nucleotide. A nucleotide is something referred to as a monomer, and a monomer is a single unit or a single building block. Okay, so the idea is if I take a unit, a single unit, a brick in the wall, right? If we take one of those bricks in the wall, that's not going to build a school. I'm sorry, one brick is not a school. But if we take a lot of bricks and we put it together, we get a school. So if a brick is the single unit, the classroom would be the combination of lots of those bricks, right? So same idea, a nucleotide is a single brick, but when we put a lot of nucleotides together, we get the classroom with bricks surrounding us. We call this a monomer, or this is our polymer. Mono, one, poly, many, right? So 1 unit, mono, poly, many. A monomer, the nucleotide is a monomer of DNA. We take a bunch of nucleotides, we put it together, we get DNA molecules. Or RNA, we get nucleic acids. There's 3 things that are made up of, that make up a nucleotide. We have a phosphate, we have a sugar, depends upon what kind of nucleic acid, DNA and RNA, there's 2 different types of nucleic acids, we'll get to RNA. So in this case, deoxyribonucleic acid, DNA, deoxyribonucleic acid, the sugar is referred to as deoxyribose, I cut that too short, is the sugar. And then we have a base, which I said out of laziness, or, sure, I'll say laziness. We're just gonna call it a base. Here they call it a nitrogen-containing base. You could call it a nitrogenous or a nitrogenous base. There's a base. There's 4 base pairs, and that's what's shown right below, is there's A's, there's C's, there's C's, and there's G's. We don't really care that you know that T stands for thymine. We don't care that you know that the T is thymine. but we would expect you to say, hey, hey, that's one of those bases, and we use T's. Okay, so just realize A's, T's, C's, and G's, and they're straight off the first letter of those words. So if we were to say guanine, that's a G, right? It's the first letter. So there's two different models that this is trying to show us. There's something called the ladder model and the helix model, and I'm not really going to talk about the helix model, but sometimes people talk about, they see that ladder model, and they're like, I don't get how that's a ladder. Well, right, this is what a ladder would look like if we're talking about like, hey, I'm gonna climb up on my roof. And when we talk about that, what we have is we have a sugar, and then we have these phosphates in between. So we have sugar, phosphate, sugar, phosphate, sugar, sugar, phosphate. Okay, and they are, okay, they kind of go through like that. And when we talk about a ladder, we call the sides of the ladder the backbone of a DNA molecule. And when we talk about the rungs of the ladder are what you physically step on. And some people say, I don't know what a rung of a ladder is. It's the part that you put your foot. So when we talk about the rungs of a ladder, we are talking about the base pairs that go there. Okay, so again, on our model, this is where I'd put my foot. These are the rungs of my ladder. These are what we'd refer to as the backbone or the sides of the ladder. Ooh, that is a nice ladder. Thank you. It's nicer than mine. Okay, so there are three, so we're not gonna do all these, but I do want to reinforce a few of these things. So we circled a nucleotide, 1A, what are the three parts of a nucleotide? We need a sugar, It doesn't have to be deoxyribose. For DNA, it is. There's something called RNA, it's a different sugar, it's called ribose. But we need a sugar, we need a phosphate, and we need a base of some sort. A nucleotide consists of three parts, a sugar, a phosphate, and a base. We expect you to know that. In DNA, the sugar is deoxyribose. If we were talking RNA, the sugar is referred to as ribose. Okay, and we'll get to RNA probably in next week, Monday and Tuesday. But in our molecule for DNA, the sugar is deoxyribose. Then I'm going to skip C, which part contains the nitrogen containing bases, but more important than that is what are the four bases? And again, I don't care if you know them by names, but letters, A, C's, T's, and G's. Okay, and A's and T's go together, and C's and G's go together. Okay? And that's what we could pull from that diagram. And some kids, some students even ask the question, like, one of the questions is asking you to like label what are the bases, and there was a little bit of confusion. If you're new to these, or maybe you've never really kind of mastered these POGL activities, everything should be pretty self-contained. There's not a lot of prior content that we ask you to do to label these. So you can say, well, how do I know which one is which? Well, the answer is you look at the key. And there's this key over here that says, hey, look, this little thing with an arrow or a tip on it, that's my A, so that's an A, that's an A, that's an A. The thing with the stripes is the T's, my T's, my T's, my T's. And if we notice that right next to every A, according to my key, there's a T, and right next to every C, there was a G. That follows something that we refer to as Chargas Rule. Okay, we're going to skip number 2, not the explanation. I'm not going to go through the explanation of why it's a ladder, but because we've kind of talked about that, right? So it's sometimes referred to as a ladder model. So just kind of be aware of that. If we were to uncoil, it's a double helix, so it is twisted. But if we were to uncoil it, it would be, we sometimes refer to it as a ladder model. All right, when we turn the page, one of the things, I guess Mr. DeBoer already did it, he circled a nucleotide on our ladder model. So this is a nucleotide, right? And our nucleotide consists of a phosphate, a sugar, and one base. Notice how he only circled the C, and he did not circle the G, because it only contains one base. Now, there's lots of nucleotides here, right? We could come over here, and we could circle a phosphate, a sugar, and a base, that would be a different nucleotide. But you need to make sure all would contain the same, it has to contain 3 parts. Now, something to note, this is not a mistake, this is very intentional. Notice how they start with a phosphate here, and there is not a phosphate here. And then on this side, there is a phosphate with no phosphate here. That's intentional, it wasn't an oopsie-doops, it was very intentional. Because what these 2 molecules do is they run in opposite directions, and they refer to that as an anti-parallel. Okay, our sugar phosphate backbones run parallel to one another, but they're running in opposite directions, so they're referred to as anti-parallel, that sugar phosphate combination. So you'll hear that from time to time. But that's what we talk about when we talk about anti-parallel. That's not an accident, it was intentional about how they drew them up. All right, so if we turn the page, when we talk about the rungs of the ladder, the things that we would step on for 3A, we'd step on the bases, the C's, the T's, the G's, and the A's. We'd step on the bases. And what parts make up the sides of the ladder? That's our sugars and our phosphates. The sides of the sugar phosphates, the rungs of the ladder, are the bases, the things we step on. The things we grab with our hands, assuming you grab the side of the ladder, not the rungs. Some people grab the rungs, so grab the side. I don't know, I guess it's a preference. Is the sugar phosphate. Okay, we're gonna skip number 4. We're gonna skip number 5. I don't care, let me just talk about number 5. I don't care if you know the difference between a purine and a pyrimine. What those are referencing is ring structures. Some are single rings, some are double rings. There's a reason to know it. It's not necessary for us. I'll be honest, I don't know the difference. I can't tell you which base is a purine and a pyrimine, because in my mind, it's not that important for my level of understanding that I need to know for DNA. So I'm not gonna expect you to know it either. Okay? However, number 6 is a very important idea. And the idea of number 6 is what kind of always goes together. And that's referred to as Chargas Rule. And Chargaff's rule says that A goes with a T, and T's goes with A, and C's go with G, and G's go with C's. Okay? That's an important thing. We are going to refer to as complementary base pairing. We're going to use complementary base pairing a lot. Base pairing, the bases, A's, T's, C's, and G's. What is the complement? What's the thing that goes along with it? The A's go with T's, T's go with G's. So that's what we're going to practice in number 7, is building the complement strand. So if we only knew one side of the DNA, right? So if we only had one side of our ladder and we knew it was TCTA on my board, we would be able to build a complement. So on this particular strand, if we were to look at one side, it's ATC, CA, GT, GA. And Chargas Rule, and we'll kind of go through the first 3 together, if the template, the original strand, is ATC, then for every A we would say there's a T, and for every T there's a and every C there's a So tag, you're it, you do the rest.
Hello.
Okay? You do the other 6. What are the complements of those bases? Using Chargas Rule. We good? Okay, so for every C there's a G. Every, I don't even know, I'm not looking.
Every A,
C,
every G,
C,
T,
A,
G,
C,
A.
T.
Bingo! You have just made The complementary strand. Pat yourselves on the back.
So what might happen if one of these complementary bases wasn't copied correctly? What do we call that? If things aren't copied correctly, we would say there is a
Mutation.
A mutation, absolutely. There was a mutation that occurred. Now, we often think of mutation being like, freaks, okay? Extra eyeballs. Not always. A mutation could be bad, could be detrimental, okay? It could be good, or it might have zero effect at all. Okay, so kind of get rid of that idea that all mutations are gonna make you, like, look really weird, or maybe even cause you to die, or because sometimes a mutation will make you grow much larger than you're supposed to and be able to fight Splinter was become like these teenage mutant ninja turtle fighting crime machines, right? Mutations were good for the teenage mutant ninja turtles because now they're like full-size people and they're heroes in a half shell. Turtle power. Okay, so not all mutations are bad. Some are good, and now in all seriousness, there are good mutations, right? If we had a mutation that if I particularly had a particular mutation that made me immune to COVID-19 or SARS-CoV-2, that would be a good mutation for me, right? So not all mutations are bad mutations. And sometimes a mutation, you wouldn't even know you have a mutation because it has zero impact on that individual. So just be aware of that. So using Chargaff's rule, we're going to fill out these, and what Chargaff found out was we were consistent with this complementary base pairings, proportions and ratios that he saw in humans was true with all organisms. So the idea again with Chargaff is A's goes with T's and C's go with G. So we're going to go through and we're going to fill this kind of quickly out together, the first one, and then we're going to have you do the rest of them just to kind of practice. So if there were 31 adenines, A's, we would have to have 31 what? T's, right? Thymines, yep. And I'm good with you calling them letters instead of the names. If you want to call them by name, you're more than welcome to. But if for every 31 A's, we have to have 31 T's. And if there are 19 C's, that would mean we'd have to have
19 G's.
absolutely, and that's what Chargas Rule says. You try the cows, and then salmon. Maybe, well, do the rest. Try the rest. We'll go through them quickly.
Spelled effect right today.
I wrote an email to someone, and I always struggle with it's the E or the A, but autocorrect fixed it for me, so it's good. Or word, like underline, like, you mean this? And like, apparently I do.
Absolutely.
Of course.
Of course I do.
Yeah. All right, so if cows have 28 A's, then they should have 28,
T's.
and if they had 22 G's, then they should have
22 C's.
absolutely. How many A's would salmon have?
29.
29. And how many G's would salmon have?
21.
21. And how many G's would wheat have?
I don't know.
Okay. How many C's would it have then?
We don't know.
But what would have to be true about this?
Zero.
Could be.
Triple
Sure. Okay, what if Mr. DeBoer wrote down 12? There were 12 G's. Then how many C's would there need to be?
12.
So what must be true about those two numbers?
They have to be the
They
same.
have to be the same, absolutely. They have to be the same. Okay, so whatever they are, you pick a number, you could go with that. Thymine, how many thymines would there need to be?
27.
Yep. And yeast would have how many C's?
16.
And how many T's?
34.
Awesome. Okay, that's Chargas Rule. So using Chargas Rule, on page 3, take that half of a DNA strand and fill in the complementary base. Only, don't worry about the sugars and phosphates right now. What is the complementary base? So for A, right above the A, you would put a T. Okay, and then you go down the rest of that line. There's 6 of them for you to decode, code. All right, so for every A, there's a T, we already said. For every G, there's a,
C.
I'm sorry, what?
C.
For every G, there's a C, okay? For every T, there's a,
A.
and for every A, there's a T, and for every C, there's a,
G.
and every G, there's a
C.
C. Okay? Now, I'm not going to ask you right now to draw the sugar phosphate because honestly it gets kind of messy, but realize that attached to the base would be a sugar, and then the sugar phosphate, sugar phosphate, sugar phosphate. Realize that. I do also want to point out once again that this starts with a phosphate and it ends with a sugar. And to follow kind of our anti-parallel model, we would start with a phosphate, then a sugar, then a phosphate, then a sugar, right? Because they run in opposite directions. Okay, just kind of keep that in mind. Last things, there's 4 quick things, and these are in your notes as well as in that activity. I believe they were in the POGL activity, but I just want to reinforce this. The bonds are held together by what kind of bond? Anybody remember?
Hydrogen.
Hydrogen bond, absolutely, hydrogen bonds. And hydrogen bonds are they have hydrogens, and it has to do with the unequal sharing of electrons. But if we talk about like water droplets, if you take like a penny and you put a bunch of drops of water, you kind of get that bubbling effect. It's because the hydrogen bonding of water that's holding it together. So why is hydrogen bond good for these base pairs? It's strong enough to hold the bases together. But a fair amount of times, these get unzipped or ripped apart. These hydrogen bonds need to be broken. So it's strong enough to hold them in place, but it's weak enough that they can be broken as needed. The kind of example, analogy that I talk about is, hey look, those really great magnets that we get in the mail, they hold up a piece of paper. But you put about 2 pieces of paper and you're about done. So this magnet serves the purpose of it holds a piece of paper on place. And it's nice because when I want that piece of paper, it's very easy for me to get up. Now, are there other stronger magnets than these wonderful ones that we get in the mail? Yeah, you've heard of like neodymium magnets, those really rare earth magnets, those really, really strong ones. Would that hold the paper on my board? Absolutely. But what happens when the time comes that I want to take that magnet off? It's hard, right? It's like, yeah, I got it. Okay? So it's strong enough. This is kind of like a hydrogen bond. It's good enough to hold it there. But if I'm going to be taking this piece of paper on and off a lot, it'd be nice to make it relatively easy. That's exactly what a hydrogen bond is. Compare that to the sugar phosphates. That's a covalent bond, it's referred to as. It's due to unequal sharing of electrons. I'm sorry, sharing of electrons. And a covalent bond it's strong. So when we go through and we replicate DNA molecules or we make mRNA molecules, we want to break that hydrogen bonds found in the middle really easily, but we want that sugar phosphate to hold tight. So we need that to be a strong bond. To kind of wrap everything up, this is kind of the ideas again that we're reinforcing that. That was really the intent, is just to kind of reinforce all these ideas. There was one more sheet that you picked up on the way in, or I'm sorry, not on the way in. There was one more sheet that you picked up when you were looking at your, well, I'm sorry, trying to navigate Canvas and talk at the same time didn't work. There's 2 things, we just did one of them. The other thing is a section 2 guided notes. That's your one of your virtual day assignments for the weekend. Your other virtual day assignment is, so virtual day assignment, there's a to-do list. There's 2 things listed that will take you to it. Otherwise, when you click on modules, you'll see this list here. There's a video, and if you had me or Mr. DeGrand last semester, I don't remember. If you had me or Mr. DeGrand, we did a couple of these last semester. If you had Mr. Lukey, I'm not sure if he did. But what we have is we have a video for you to watch and a Google form questions for you to answer. So there's a video linked here, it's also linked here, there's a Google form here, there's also the Google form here. But when you click on the Google form, there's again the link, there's the video that we've embedded. There's a series of questions for you to answer. When you are done, you hit submit, and then we want you to take a screenshot of your score and submit the image of your score.
Your score.
Your score. Okay, it'll score it for us. You submit your score.
Your score is your grade.
If you get 5 out of 10, That's a 50%, right? That's not great. It's even debatable whether it's an LP or an NE at that point. If you get a 10 out of 10, I think we would all agree, hey, that's an A. So the grade we enter is going to be reflective upon the thing you take. So my suggestion, if you have the capability, would be kind of do 2 different devices, one with the video, one with the questions. And as you're watching, I think it's a 12-minute video, you're kind of looking for those questions as you go, and then when you're done, you can kind of hit submit. That'd be my suggestion, but we can absolutely make it work with one device as well. Those are your virtually assignments for the weekend. We will see y'all on Monday or Tuesday, you guys Tuesday, but maybe y'all are watching from YouTube land. So have a great weekend, everyone.
Why are these 100 watching daily?
No.
Ready for?